Hello students, I hope you must be having a wonderful day. Let us discuss about the few objective questions that are concerned with the forensic biology. First question is about blood grouping. If a father has blood group genotype AO and mother's blood group genotype is BO. Okay, AO is father's blood group genotype and mother's blood group genotype is BO. So here we are asked to figure out what will be the possible blood group genotype of the offspring. First is either it could be AB blood group or it could be AO, then it could be BO genotype or simply OO genotype. Now we know that O is recessive whereas A and B are dominant. So here are probabilities that child could have AB blood group or A blood group or B blood group, but there is no or very least probability of child to have O blood group. O blood group is not mentioned in the option, but we obtained results as AB and AB, hence the option D would be the correct answer for the respective question. Now in old blood stained fiber, blood grouping is done via, simply the question mean to ask you that what is the method that is used for blood grouping of an age old stain. So the answer would be absorption elution test, because the benzidine test is a presumptive test and so is precipitin test. Hemin crystal is one of the confirmatory tests. It is used for the in order to identify whether the obtained fluid is blood or not. It has no relation with blood grouping. Fine. Which of the following solvent indicate that the menstrual blood was ante-mortem? Ante-mortem means before death. This question mean to ask you that how will you be able to identify that the blood that you have found is from a woman who probably would be menstruating before her death. So here's the answer. That blood would not show clot unless you mixed it with seminal fluid. Okay, if there is no clot present in the blood, it means that it must be the ante-mortem blood. Now the Takayama test, it is a confirmatory test. Takayama reagent is used in so in case you know, Takayama test is also known as hemochromogen test. So this Takayama reagent is used in hemochromogen test, whereas Hemin crystal name is another name given to the Teichmann crystal test. Now which of the following is wrongly matched? Here's the test and here are the color that possibly could be obtained from the respective test. So yep, benzidine test yields blue color, Gucum test gives deep blue color, that is true as well, and here you will get pink needle sheep crystal and Casimir test. But here's the twist, for leucomalachite green you won't get pink color, you would get some bluish to green color. Okay, I guess the next question is continuation of the previous question. Leucomalachite test, it is a kind of presumptive test or screening test. This test gives you peacock blue color. Fine. Then best method to prove paternity is blood grouping, that is of no use. Fingerprinting is not even of much use to prove paternity, so as this phenotypic resemblance. So yep, the answer is DNA fingerprinting. This is the best method known so far in order to prove the paternity. In Takayama test, which of the following is seen under the microscope? As I have explained to you that Takayama is a confirmatory test. It is done for blood. So the answer is pink feathery crystal. Pink feathery crystal indicate that the fluid that you were performing test on is blood. Now moving forward to the 9th question. Again some same kind of question that we have already discussed for paternity. It is DNA fingerprinting. Species identification is done using, in case you know that spectroscopy is not of much use, benzidine is presumptive test of blood. You can't expect that benzidine test would help you to identify whether the sample belongs to Homo sapiens or any other species. So the answer would be precipitin test. Na is useless in this case as well. I hope you have liked this objective question series. In case you want more objective questions on different topics of forensic science and criminology, so please let me know in the comments below. Thanks a lot.